Mendelian patterns of inheritance, lecture 2. All right, so last time we were talking about the genotype and the phenotype. These are terms now that you should have a basic understanding of, and we went over the principle of dominance. So let's look at now, dive into it a little bit more. Molecular genetics. Molecular genetics is the study of the chemical makeup of a chromosome and their genes. So before we kicked off this whole entire chapter, we looked at chromosome structure and some chromosomal abnormalities. That would come down to molecular genetics. And we talked about how those diseases, whether it be a deletion, addition, inversion, or a translocation of those genes and the parts of those genes, how they could affect the human individual. So where the genes are found at specific location on chromosomes, these are points called the gene locus, or you could say the gene loci if you're talking about many genes. So if you look, the allele for flower color, the gene for flower color, there's the locus point on the homologous chromosome pairs, and there you can see on the one homologous chromosome, you see the allele for purple flowers, that would be the capital P allele. And then on the other homologous chromosome, you see the lowercase p representing the recessive allele for white flower coloration. So here you can see examples of gene loci and particular genotypes of being homozygous, dominant, homozygous, recessive, and heterozygous. And you could see the difference between what a dominant allele would look like, as in the capital B and the capital P's here, versus recessive alleles, which would be lowercase a, and lowercase b. So here we have a homozygous dominant genotype. Here we have a homozygous recessive genotype. And here we have that heterozygous genotype, which is a dominant allele with a recessive allele. Mendel's law of segregation. So Mendel's second law, or first law, states that each individual has two factors for each trait. Now we know that these two factors are represented by the alleles. And these factors or alleles are going to separate during the formation of gametes, gametogenesis or meiosis, so that each gamete contains only one factor from each pair of the factors. That would be at the end when you have your 4 haploid cells. So each gamete gets one copy of the gene. And then upon fertilization, it gives each new individual 2 factors for each trait. So when the sperm meet the egg, you restore that diploid number. So each gene then will have those 2 factors, those 2 alleles represented for that gene. So how do we do genetic crosses to represent all this? We do it through using Punnett squares. And a Punnett square is a chart that is used to illustrate the possible outcomes of offsprings within a genetic cross. Now, as we get into this next section of the lecture, I'm going to go over it here, discuss it here on the lecture presentation. However, in class, we will go over multiple examples of illustrating Mendel's laws, the principle of dominance, the genotypes and phenotypes, and doing genetic crosses using Punnett squares. So this will be a kind of verbal introduction, and then we'll do hands-on stuff in the classroom setting. So genetic crosses. Keep in mind that as we do genetic crosses, we do need to apply statistics here. Our first type of cross we're going to look at is a monohybrid cross, and monohybrid crosses are genetic crosses that involve only one gene. And then we're going to do a test cross, and we'll discuss what a test cross is all about. And then a dihybrid cross is a genetic cross that involves 2 genes. It's a 2-gene cross. So monohybrid cross, here you can see the cross between the purple true breeding white flower pea plant versus the true breeding purple or white flower pea plant versus the true breeding purple flower pea plant. Look at the gamete formation there. Because of the law of segregation, each gamete could only contain one copy of that gene. So there in the gametes you have the capital P for the purple flowers, the lowercase p for the white flowers, and when those are fertilized, those gametes meet, you have an F1 generation that has all purple flower coloration. However, what you have there is genotypically all heterozygous offspring, phenotypically all purple flowers, because this is a case of complete dominance where purple dominates over white. If you do an F1 cross, so you cross two heterozygous purple flowers from the F1 generation, your F2 generation results in 3 purple flowered pea plants and 1 white flowered pea plant. So let's look at that there. Quick discussion. Because they're heterozygous, you have a half chance or 50% chance to pass on the dominant allele, capital P, to the gamete, or you have a 50% chance to pass on the recessive lowercase p, the white allele, to a gamete. So you have to account for all possible combinations there. So when you set up the Punnett square, you're going to have, for one parent, you're going to have big P, little p. For the other parent, you're going to have big P and little p, as shown here. Then when you cross it, it's kind of like playing Battleship, where you drop down the alleles for that cross. Where did my pointer go? And there you have big P, big P. Here you have big P, little p. Here you have little p and a big P. And here you have a little p and a little p. So you have 3 purple flowers, 1 white flower. So the phenotypes ratio, the phenotypic ratio would be 3 to 1. The genotypes, the genotypic ratio would be 1 to 2 to 1, because you have 1 big P, 2 big P little p's, and 1 little p. And we'll go over that a lot more in class. We'll do several examples that will help illustrate this a little bit better. So there you can see the phenotype, 3 purple, 1 white, so that ratio there is 3 to 1, and the genotypic ratio would be 1 to 2 to 1, and there you can see it there. So here is a chart illustrating the dominant trait and the recessive traits for flowers, flower position, seed color, seed shape, pod color, stem length. You can see that purple is the dominant flower color to white, axial is the dominant flower position to terminal, yellow is the dominant seed color to green, round is the dominant seed shape to wrinkled, inflated is the dominant pod shape to constricted, green is the dominant pod color to yellow, and tall is the dominant stem length to dwarf. Now, a test cross. A test cross. How can we tell the genotype of an individual that expresses a dominant phenotype? Basically, what we need to know here is that the phenotype is dominant, therefore one dominant allele must be present. So you have a dominant phenotype, therefore your genotype could be homozygous dominant, or it could have the heterozygous genotype. And to figure out what the unknown genotype is, you could perform a test cross. And we'll go over an example of that. Basically, what we have here is, of this parent here, that is the purple flower parent, the dominant phenotype, unknown genotype. We know that the dominant phenotype is purple. We don't know whether it's a homozygous dominant genotype or a heterozygous genotype. So what you're going to do is you're going to cross that with a recessive phenotype. And therefore we know that that genotype of that parent will be little p. So if big P, then all the offspring would be purple. So you'd be crossing a big P crossed with the other flower being little p. We'd have all heterozygous genotypes there, and all the phenotypes expressed to be purple. If it were the other scenario, where you have the heterozygous genotype for the purple flowered plant crossed with the homozygous recessive white flower plant, then you'd end up with 2 purple flowered plants among the F1 offspring and 2 white flowered plants among the F1 offspring. So you'd have 50% purple flowers and 50% white flowers, and those purple flowers would have that heterozygous genotype. And we'll go over examples of test cross in class. A dihybrid cross involves a cross between 2 true breeding parents differing in 2 characteristics. Mendel's dihybrid crosses illustrated his second law of inheritance, which is the law of independent assortment. So what does the law of independent assortment state? It states that each pair of factors, these would be the alleles, so each pairs of alleles are going to segregate or assort independently of other pairs of alleles during gamete formation to allow for all possible combinations of factors occurring in the gametes. So when we talk about in meiotic cell division, we talked about increasing genetic variation. Here is another way to account for all possible variations seen among the offspring and in those gametes formation, because you have crossing over occurring. Here you have independent assortment, and we know that the alleles for an individual gene are going to segregate independently of one another. So they're going to segregate to ensure that each gamete gets one copy of the genes. And now we know in the law of independent assortment, it states that each pairs of factors segregate or sort independently of other pairs of alleles during gamete formation, gametogenesis. So this all accounts for that increase, takes into account for increased genetic variation. So this applies only to genes on different non-homologous chromosomes. So basically what it's ruling out is if you have, if you think of the 46 pairs of chromosomes and you think the non-homologous pairs, it states that, you know, if the homologous pairs of one chromosome, if the dominant allele goes to one gamete, then that doesn't necessarily mean that on another non-homologous chromosome pair that is different from the one that already separated, the dominant alleles are also going to go and follow the dominant alleles of the other homologous chromosome. What this states is that you can mix and match to have all possible combinations, and the genes located near each other on the same chromosomes tend to be inherited together. So the genes that are close together on one chromosome will follow that chromosome and go into that same particular gamete. So here is the hypothesis of dependent assortment, and basically you can see it illustrated here that during gamete formation the dominant alleles don't follow one another and the recessive alleles do not follow one another. So the hypothesis of dependent assortment contradicts what really happens is this, where you need to account for all possible gamete allelic combinations. So you could have big R, big Y, little r, you could have big R, little y, and you could have little r, big Y. So you have to account for all possible combinations and take into account all possible scenarios among the offspring. And we'll do a dihybrid cross example in class. So frequency of dominant alleles, to kind of wrap up the case of complete dominance, dominant alleles are not necessarily more common in a population. 1 in 400 babies in the United States are born with extra fingers or toes, so that extra digit. The allele for the extra digit is dominant to its 5-digit counterpart. And that's where we're going to end this lecture for today, because so far up into this point when we did monohybrid crosses and we did dihybrid crosses, and our entire discussion kind of was centered around the case of complete dominance. So when that dominant allele is present, it will completely override that recessive allele. However, our next section of the lectures, lecture 3 of Mendelian genetics, is going to go beyond dominance. So we're going to do some special case scenarios and look at what that means for the organism and further elaborate and educate ourselves in the field of genetics, because it's not always this simple of the case of complete dominance. There are many other factors that play into it. So we'll get into that in our next lecture.